All right, so our next topic for discussion is going to be phagocytosis. All right, we talked about the cells of the immune system that are important in phagocytosing, the process of phagocytosis. Those were the neutrophils and the monocytes, specifically those macrophages and dendritic cells. Those are all going to be phagocytes. All right. Now, one of the key things with the immune system that we will talk about a little bit now and a little bit more in chapter 17 is the concept of self versus non-self, okay? Because you don't want your neutrophils or your macrophages to start engulfing and breaking down the cells of your liver or your nervous system or your kidneys, okay? So those cells that have that ability to undergo phagocytosis have to have a way to recognize what belongs and what doesn't, okay? So essentially what will you have with the recognition of self versus non-self are going to be markers, or really what we're talking about are going to be proteins that are found on the outside of a cell, all right? So in terms of that bacterial cell, there are going to be proteins, and that's what the lines are that I drew on there, that are going to be foreign to your immune system. Whereas with the eukaryotic cell within your body, it isn't going to have that marker on the outside of it. So in terms of that protein, all right, kind of a catch-all classification for that is a pathogen associated molecular pattern, or PAMPs, okay? So you have these markers that are on the outside of pathogenic cells. This is going to be what the immune system recognizes. All right. So in terms of that, that's one of the keys that the immune system is going to pay attention to recognize self versus non-self, okay? There's more involved with it, and I'll just very quickly kind of put this up here. You've got what's known as major histone compatibility complex. This is essentially a tag that's on the outside of all of your eukaryotic cells that make you up. That's essentially the secret handshake with the immune system, all right? Or the secret pass, if you want to think of it that way. What happens? The immune cell comes up, that phagocyte, whether it be a macrophage, whether it's a dendritic cell, whether it's a neutrophil, it comes up to that cell. It feels the cell surface. If it encounters the major histone compatibility complex, it's going to be like, oh, you're one of us. You get to stay, all right? If that macrophage comes up and senses then our pathogen molecular marker, the PAMP, that's going to be the signal for that phagocytic cell to now go and engulf that bacteria. All right, and you'll have to forgive me, this is not drawn to size. The bacteria is not going to be bigger than the macrophage. That's just a limitation of space, okay? But in terms of that process, one of the components that's important with phagocytosis is going to be the process of chemotaxis, all right? If you look at the word chemo, that signifies something chemical. Taxis indicates movement. essentially what happens, those phagocytic cells, the macrophage, it has the ability to sense various chemicals, like the proteins on the outside of that bacterial cell. That's going to cause that macrophage to move towards that particular pathogen, all right? And I have a video up that I put up of a neutrophil chasing the yeast cell. That's the same idea. That's that process of chemotaxis. If you watch that neutrophil, it's kind of feeling around the environment, and then it latches onto that yeast cell, and it goes in for the kill, and it engulfs it, and it'll start breaking it down, okay? So in terms of phagocytic cells, that ability to undergo chemotaxis is going to be important, all right? Now in terms of that recognition, all right, we've got our pathogen-associated molecular marker here. I should have drawn this a little bit better. We'll put this in blue here, okay? We've got the toll-like receptor that's on the outside of our macrophage, okay? And I'll label this here. This is our macrophage, okay? This is our bacteria, all right? In terms of that, it's going to be the toll-like receptor that's going to recognize the pathogen-associated molecular pattern. That is then going to be what targets that process or the start of phagocytosis. Because when that toll-like receptor recognizes the bacteria or the PAMP, it's going to trigger the release of something known as cytokines that we haven't talked about yet, all right? But cytokines are another way for the immune system to talk to itself, to talk to the other components. When cytokines are released, typically what that does is it triggers one of two pathways. It'll trigger phagocytosis, but the release of those cytokines from that macrophage is also going to be important in starting the inflammation response, okay? And that's what the next video will be. For now, we're going to focus on phagocytosis, okay? Because in terms of phagocytosis, you're going to have some different stages associated with phagocytosis. You're going to have the chemotaxis first. You're going to have that macrophage or neutrophil find the bacteria, okay? It's going to have to attach. And this is where our toll-like receptor comes into play, all right? And then what's going to happen, that macrophage is specialized for engulfing, all right? So if you kind of think of it as targeting its prey, it's going to wrap that cell membrane around our bacteria, where that bacteria then can be broken down, okay? And in terms of breaking down the bacteria, you're going to have another component of that eukaryotic cell that's going to be important with that. You're going to have the lysozyme that's going to fuse with that engulfed bacteria, and it's going to break it down, all right? So if we use our cell over here, all right, now we're going to make this our macrophage, okay? Because I want to kind of blow things up here, or at least draw it so you get a better feel for things, okay? And in terms of that macrophage, all right, it's going to form a little vessel that now is going to contain our bacteria, all right? We're going to have the lysosome, that's going to fuse with that vessel. And what it's going to do, Let's make our enzymes orange. Oh, I don't know if you can see the orange. That's okay. All right. Let's make them blue. All right. The blue is going to be now our lysozymes. Once that fuses, and we now have our bacteria with those lysozymes inside of it, that's now going to target that bacteria, and it's going to break it down into little bitty pieces of protein. Okay, so it's essentially going to destroy the bacteria. All right, so once that lysosome fuses, you get the destruction of the particular pathogen. I've drawn a bacteria here. It could also potentially be a virus that's the pathogen. Okay, but in terms of that, you've got that toll-like receptor on the outside that's going to recognize, bring in our pathogen. Once inside, that pathogen is going to be broken down by enzymes contained within the lysosome. Once that's broken down then, all right, one of two things can happen with the macrophage. I'm running out of space, all right? Either that vessel then will fuse with the cell membrane and you'll get rid of those bits of waste. Or when we get to chapter 17, I'm going to talk about this in more detail, you can have those particles essentially displayed on the outside of the macrophage. Okay? Now keep in mind this pathway here is going to be specific for macrophages and dendritic cells. Okay? Macrophages and dendritic cells have the ability to display parts of that bacteria on the outside of their cell membrane to bring in the specific immune response, specifically either B cells or T cells. Okay? Your neutrophils are always going to go the route of eliminating the waste. Okay? So in terms of that, the last step there, elimination of waste. That's primarily going to be the neutrophils that are going to take that pathway. Macrophages and dendritocytes, because they're antigen-presenting cells, this is what it means to be an antigen-presenting cell. You're presenting bits of that infectious material on the outside of the cell membrane. Now when that macrophage or that dendritic cell interacts with a natural killer cell, with a B cell or a T cell, that's going to trigger a more specific immune response. Okay? Don't worry about that part right now. All right? I just want to kind of introduce you to that particular concept. We're going to go into more depth in it with chapter 17. All right? But I do want you to know the stages of phagocytosis, what's happening at each of the different stages. All right, and essentially this is part of your innate immune response for disposing of pathogens that get in. Okay, the macrophage, the neutrophil, the dendritic cell, there's no specificity to what they recognize and engulf. They may put on the outside of their cell membrane something that's pathogenic. They may put pollen out there, which is not dangerous. All right, so in terms of that, keep that in mind. If you have any questions, please let me know.